Gene cloning. Gene refers to any part of DNA, and cloning means multiplication. So, gene cloning can be defined as a process in which a gene of interest is located and copied out of DNA extracted from an organism. That organism can be anyone. So, moving towards the procedure, it contains three major steps. These are selection of gene of interest, vector selection, and transformation. First of all, we have a bacterial cell. It contains our gene of interest. We have to extract the DNA of bacterial cell, and we put this extracted DNA into a test tube. Let us say that this is our test tube number 1, and a specific restriction enzyme is added into this test tube. This restriction enzyme will cut our gene of interest into smaller pieces. Then a plasmid is taken from another bacterial cell, and it is again cleaved in the same way with the same specific restriction enzyme. What happens when these cleaved plasmid is added into the gene cut sized pieces into the test tube? Those genes will join with our plasmid, and this joining will lead to the production of recombinant plasmid. A recombinant plasmid contains gene of interest, and every plasmid, as we already know, that naturally contain an antibiotic resistant gene. Now bacterial cells are added into the recombinant plasmids, and these bacterial cells will uptake the plasmid and become transformed. How this is going to happen? We have to perform the electroporation. In electroporation, the electrical pulses will produce holes in the cell walls of bacterial cells, and recombinant plasmid will easily enter into the bacterial cells through those holes. Then not all the bacterial cells are transformed. So these bacterial cells are grown into the separate petri dish that contains a antibiotic, as any antibiotic, and only those bacterial cells which are transformed by uptaking the recombinant plasmid will be able to survive, as they will show resistance to the antibiotic, and the others will die. So we separate our transformed bacterial cells and grow them separately to get an expression of gene. Now let us look at this diagram to have a better idea about how gene cloning actually works. We have a bacterial cell that contains our gene of interest, and this gene of interest is represented by the highlighted area. We extract this DNA from the bacterial cell and put it into the test tube. Let us say that this is our test tube number 1. We add a specific restriction enzyme into the test tube so that it can cut the gene into smaller pieces. This restriction enzyme will produce smaller segments of gene with specific sequences. We know that all the bacterial cells naturally contain an extra chromosomal DNA, and this DNA is called as plasmid. This plasmid is extracted from bacterial cell and added into another test tube. Let us say that this is our test tube number 2, and the same restriction enzyme is used to cleave our plasmid. These cleaved plasmid have specific sequences of nucleotides which have affinity to join with our gene of interest. However, not all the plasmids join with gene of interest. Some plasmids can join with themselves in absence of gene of interest, and this leads to self-ligation. Then this cleaved plasmid is added into the test tube number 1, and the gene which was cut into smaller pieces will join with this plasmid, and this leads to the production of recombinant plasmid. The recombinant plasmid contains our specific gene of interest and an antibiotic resistant gene which is naturally present in almost all plasmids. Now bacterial cells are added into the test tube that contains our recombinant plasmid. These bacterial cells can take up this recombinant plasmid, but not all the cells can uptake the recombinant plasmid. Electroporation is done to enhance their ability of transformation. The transformed bacterial colonies are separated from non-transformed bacterial colonies and grown separately to get the expression of gene.